We are going to talk about the physiological importance of stomach. Stomach performs the mechanical function as it helps in the storage of food. Food is stored in the stomach for a long period, that is for 3 to 4 hours, and emptied into the intestines slowly. The maximum capacity of stomach is up to 1.5 liter. Slow emptying of stomach provides enough time for proper digestion and absorption of food substances in the small intestine. Another mechanical function of stomach includes the formation of chyme. Peristaltic movements of stomach mix the bolus with gastric juice and convert it into the semi-solid material known as chyme. Other than mechanical functions, stomach also performs the digestive function, which refers to the function of gastric juice. Gastric juice acts mainly on proteins, and the proteolytic enzymes of the gastric juice are mainly pepsin and renin. Gastric juice also contains some other enzymes like gastric lipase, gelatinase, urase, and gastric amylase. The role of pepsin is that pepsin is secreted as inactive pepsinogen. Pepsinogen is converted into pepsin by hydrochloric acid, and the optimum pH for this activation of pepsinogen is below 6. The action of pepsin is that pepsin converts proteins into protoses, peptones, and polypeptides. Pepsin also causes curdling and digestion of milk. Gastric lipase, that is another enzyme in gastric juice, also plays a role in digestion, as gastric lipase is a weak lipolytic enzyme when compared to pancreatic lipase. It is active only when the pH is between 4 and 5. Gastric lipase is a tributyranase, and it hydrolyzes tributyrine, butterfat, into fatty acids and glycerols. In addition to mechanical and digestive function, stomach also performs hemopoietic function, which involves the role of intrinsic factor of Castle secreted by parietal cells of gastric glands. They play an important role in erythropoiesis. It is necessary for the absorption of vitamin B12, which is called extrinsic factor, from GI tract into the blood. Vitamin B12 is an important maturation factor during erythropoiesis. Absence of intrinsic factor in gastric juice causes deficiency of vitamin B12, leading to pernicious anemia. Other than this, stomach performs protective function as well, which refers to the function of mucus. Mucus is a mucoprotein secreted by mucous neck cells of the gastric glands and surface mucus cells in fundus, body, and other parts of stomach. Mucus protects the gastric wall from irritation or mechanical injury by virtue of its high viscosity. It prevents the digestive action of pepsin on the wall of the stomach, particularly gastric mucosa. It also protects the gastric mucosa from the action of hydrochloric acid because of its alkaline nature and its acid-combining power. After learning about all main roles of stomach, let us talk about the secretory function of cells in gastric glands that are mainly chief cells, cell in stomach, that have secretory products listed as pepsinogen, renin, lipase, gelatinase, and urase. The parietal cells have secretory products named as hydrochloric acid and intrinsic factor of Castle. And the mucous neck cells have a secretory product called as mucin. These stomach secretions collectively form digestive fluid that is commonly termed as gastric juice or gastric fluid. Gastric juice is a thin, colorless fluid which promotes digestion of the stomach. The composition of gastric juice comprises of water, whose percentage is 99.5%, and solids, which are 0.5% of total composition. The solids are further divided into organic and inorganic substances. The organic substances consist of enzymes and some other organic substances called as mucus and intrinsic factors. The inorganic substances include the hydrochloric acid, sodium, potassium, and calcium. Well, we are aware that digestive enzymes make a great portion of gastric juices, and we already briefly discussed them under digestive role of stomach. The first enzyme here is pepsin, whose activator is HCl. Its substrate is protein, and the end product is protoses, peptones, and polypeptides. The second enzyme is gastric lipase, whose activator is acidic medium. Substrate is triglycerides of butter and end product is fatty acids and glycerol. The third enzyme is gastric amylase that activates is acidic medium. The substrate is starch and the end product is dextrin and maltose. The fourth enzyme is gelatinase, whose activator is acidic medium and substrate is gelatin and collagen of meat and end product is peptides. The last enzyme of gastric juice is urease. The activator is acidic medium. The substrate is urea and the end product is ammonia. After this, let's look at the control of these gastric secretions. Gastric secretions occur in basically three phases: cephalic phase, gastric phase, which is the second one, and the last one is intestinal phase. Starting with cephalic phase, secretion of gastric juice by the stimuli arising from head region, cephalus, is called cephalic phase. This phase of gastric secretion is regulated by nervous mechanism. The gastric juice secreted during this phase is called appetite juice. The quantity of the juice is less, but it is rich in enzymes and hydrochloric acid. During this phase, gastric secretion occurs even without the presence of food in stomach. Nervous mechanism regulates cephalic phase through two types of reflex actions. 1. Unconditioned reflex, which is inborn reflex. 2. Conditioned reflex, which is acquired through previous experience. Right here we can see the conditioned and the unconditioned reflexes. The conditioned reflex activated at the sight, smell, thought and hearing about food. And the unconditioned reflex occurs because of presence of food. It leads to the stimulation of vagus nerve that causes the secretion of gastric juice. Postganglionic parasympathetic fibers in the minteric plexus cause the release of acetylcholine and stimulate the release of gastric juices from the gastric glands. Vagal stimulation also causes the release of a hormone called gastrin from cells in the antrum of the stomach called G cells. Gastrin is then secreted into the bloodstream. Next comes gastric phase. Secretion of gastric juice when food enters the stomach is called gastric phase. This phase is regulated by both nervous and hormonal control. It causes the stimulation of vagus nerve, local nerves and gastrin, which ultimately leads to the secretion of gastric juice. The main stimuli for this phase are the distension of the stomach and the chemical composition of the food. Mechanoreceptors in the stomach wall are stretched and set up local minteric reflexes, and also longer vagovagal reflexes. Both cause the release of acetylcholine, which in turn stimulates gastrin, histamine, enzymes, and mucin. Whole proteins do not affect gastric secretions directly, but their breakdown products, such as peptides and free amino acids, do so by directly stimulating gastrin secretion. A low pH more acid in the stomach inhibits gastrin secretion. Therefore, when the stomach is empty or after food has entered stomach and acid has been secreted for some time, there is an inhibition of acid production. However, when food first enters the stomach, the pH rises, less acid, and this leads to a release of the inhibition and causes a maximum secretion of gastrin. Thus, gastric acid secretion is self-regulating. The gastric phase lasts for about 3 hours. The last phase is intestinal phase. Intestinal phase is the secretion of gastric juice when chyme enters the intestine. This phase is also regulated by nervous and hormonal control, and it starts when chyme comes in intestine. It causes the release of gastrin, which causes stimulatory effect on release of gastric juice. This phase also causes release of secretin, cholecystokin and pancreosomin, somatostatin, gastric inhibitory peptide, and vasoactive intestinal peptide, and it causes inhibitory effects on release of gastric juice. When chyme enters the intestine, initially, the gastric secretion increases, but later it stops. The intestinal phase comprises of 2 stages. In the initial stage, chyme that enters the intestine stimulates the duodenal mucosa to release gastrin, which is transported to stomach by blood. There it increases gastric secretion. During the later stage, after the initial increase, there is a decrease or complete stoppage of gastric secretion. Gastric secretion is inhibited by two factors: enterogastric reflex and gastrointestinal hormones. Enterogastric reflex action is mediated by mesenteric nerve plexus and vagus nerve, while the gastrointestinal hormones action is mediated by secretin and cholecystokinin. In human beings, a fourth phase called interdigestive phase exists. Secretion of small amount of gastric juice in between meals or during period of fasting is called interdigestive phase. Gastric secretion during this phase is mainly due to the hormones like gastrin. The gastric juice secretion is sometimes also stimulated by alcohol intake and caffeine. So, this was all about the physiology of stomach. In this section we learnt about functions of stomach, secretory functions of stomach cells, and composition and control of gastric secretions. This video must have improved your knowledge. For more such videos, keep watching sqadia.com.